Okay, so welcome back to this next video in which we are discussing endothelium-dependent vasodilatation. Okay, so I apologise for the abrupt ending to the previous video. Okay, so we're just in the process of discussing the structure of the nitric oxide synthase-free homodimer. Okay, so we were discussing the fact that it is divided up into these two key domains, the oxygenase domain, which is at the amino terminal, and then the reductase domain, which is at the C terminal here, which has got these two special portions that I've separated here. And we were just about to draw another one here, and this is the problem with my picture, that this doesn't have enough space to draw the exact opposite of this. Okay, so this one's going to have to be a much more squashed portion. Okay, so here is the equivalent of that portion there, and then here is the equivalent of that portion there. Okay, and then there is the C terminus. Right, so let's talk about some of these domains. So let's start with these oxygenase domains here. These oxygenase domains are going to have a prosthetic heme group bound to them. Okay, so I'll draw this in here. So remember, prosthetic heme groups consist of a porphyrin ring with an ion, which is a tongue twister, right at the centre. So an ion cation, maybe is a better way of saying it, right at the centre. So I'm just going to show the prosthetic heme group sticking on like that as a square, because the porphyrin ring is roughly a square. Okay, so both of these groups are heme groups. So each of the oxygenase domains has this heme group attached on. Okay, and heme groups are described as prosthetic groups, okay, because they have been stuck onto the side of the protein. Okay, so heme prosthetic group. Okay, right. Now, also bound to the oxygenase domain is a cofactor for the function of the enzyme, which is known as tetrahydrobiopterin. Okay, so I'll draw this here. So symbolically it's written H4B, like so. The full name for this is tetra, okay, which means 4, hydro, which refers to the 4 hydrogen atoms here, and then biopterin. Okay, now this is a cofactor. It's an essential cofactor for the function of the eNOS enzyme, okay, but what exactly it does is still a little bit of a mystery, okay, but it is there. So this is tetrahydrobiopterin. So those are the two important bits that are bound onto the oxygenase domain there. You have the heme group and the tetrahydrobiopterin. Let's now talk about the important bits that are attached onto these two portions here. So this portion here, this portion of the reductase domain that's closer to the oxygenase domain, this binds something known as flavin mononucleotide, and I'm only going to show this for this one where it's bigger, not for this one, otherwise it'll be a squash and I won't be able to fit it all in down there, but this one would be the same as this one here. Okay, so let's highlight this portion up in a different colour. So we'll have it up in orange here, and this portion here is the same. Okay, so this is bound FMN, which is short for flavin mononucleotide. Now, flavin mononucleotide is another electron carrier. It's capable of accepting electrons and carrying them. Okay, so we've got an electron carrier bound to that portion. Okay, then here we're going to actually have another electron carrier here, which is going to be FAD. Okay. So I'm just going to highlight this up. We'll have it in green over here. So this green portion, which is the portion of the reductase domain closest to the C-terminal tail here, this is FAD. Okay, now this is also another electron carrier. Okay, so this is flavin, that's the F, and then the AD is again for adenine dinucleotide. So again, these are very related to NAD and the NADP that we saw earlier. So again, that's an electron carrier. And then finally, next to the flavin adenine dinucleotide binding site, you actually have a binding site for NADPH. Okay, so this is where your electron donor is going to come in and bind there, and effectively what then happens is an electron transport chain. The electrons go from the flavin adenine dinucleotide to the FMN, and then they don't go to the heme group on the oxygenase domain of the same NOS-free protein here, but they go to the heme group of the opposite oxygenase domain, and then they'll be provided into the reaction. Okay, so this is an electron transport chain. Here's where you bind the input molecules, and then they're transferred along these other electron carriers that are bound to the enzyme. Okay, so this is the enzyme in the inactive state at the moment. Now, as I say, the electrons cannot be transferred from this FMN to the heme group on the oxygenase domain of the same protein. Okay, they're not close enough. And this is why these enzymes have to function as a homodimer, because this reductase domain here is actually going to function with the oxygenase domain of this other NOS-free protein here, and this reductase domain of this NOS-free protein here is going to function with the oxygenase domain of this one here. Okay, so this is actually going to be two separately functioning enzymes, but they require each other to function, basically. Okay, so they have to homodimerize, but it's not just one enzyme, this is two separate enzymes, it's just they do have to homodimerize to actually be functional because of this fact that the reductase domain of one interacts with the oxygenase domain of the other to actually make an active enzyme. Okay, so what's going to happen then is, this is it in the inactive state. Calcium calmodulin complexes can bind to these linker regions and cause a change in conformation that brings this portion close to this oxygenase domain of the other one. Okay, so let me show this on another page. Okay, right. So let's now draw out the eNOS, the endothelial nitric oxide synthase, in this now active state where it has got calcium calmodulin complexes bound to it. So let's draw the thing out again. So here is the oxygenase domain of the one on top. Okay, and I'll try and draw these a little bit smaller than before. Okay, so here are the two oxygenase domains. Here's the N-terminal of the one on top. Here's the N-terminal of the one on bottom. Now what's going to happen is, because of calcium calmodulin complexes binding, this linker has changed conformation so that the reductase domain of this one on top is now closer to the oxygenase domain of the one on bottom. Okay, so here is this reductase domain of the one on top. And the same thing has happened to this one up here, which is on the bottom. Okay, its reductase domain is now closer to the oxygenase domain of the one on top. Okay, and this is now a functional enzyme here, and this is now a functional enzyme here. So let's colour these portions in again. So here, let me just get the highlighter in the right orientation. These are the two oxygenase domains which are dimerized together here. Okay, remember we had the two heme groups here, one on this one on this one. We also had the tetrahydrobioterin cofactor present here and also present here. Okay, then this domain here, which is a part of the reductase domain, which has flavin mononucleotide bound to it, has now moved in position so that it's very close to the heme group of the oxygenase domain of this other NOS-free protein here. Okay, and then we have the other portion of the reductase domain here, which, remember, had the flavin adenine dinucleotide bound here, and then also would have the NADPH, which is providing the electrons into this transport chain. Okay, and now the key thing that I need to draw on this picture is the calcium calmodulin complex here, and another one here, which has caused this change in conformation in these NOS-free proteins that has now made the enzyme functional. Okay, so to once again stress this, this here, this is a single functional enzyme here, and this one here, this is another single functional enzyme. So actually now what you have is two separate functional enzymes there in this homodimer. Okay, right. So that's how these enzymes need to dimerize together to function, but you do form two separate functioning enzymes when you do make the homodimer. Okay, right. So now let's talk about the electron transport chain. So what happens is the NADPH initially transfers the hydride anion onto the flavin adenine dinucleotide, and then what happens is the flavin adenine dinucleotide transfers gradually single electrons to flavin mononucleotide and then gets rid of the proton into free solution. and then the flavin mononucleotide will transfer that electron to the heme group, and it's at this oxygenase domain here that the electrons are then put into the reactants, basically, and put into the reaction. So this is how the enzyme is transferring these electrons that are going to be put into this reaction at each stage. So remember, 2 electrons are being put in here, and 1 electron is being put in this at this second stage. Okay, so this is the electron transfer, and it's at the oxygenase domain that the reaction is actually going to be catalyzed. Okay, right. So that's then how nitric oxide synthase 3 works. Okay, so it will be producing nitric oxide. So now let's talk about what this nitric oxide is going to do. Okay, and once again I'm going to stress this point that I've stressed so many times now, but which is so important, that it's the not just the endothelial cells of the arteriole that are producing the nitric oxide, but also the endothelial cells of the capillaries and the venules, and it is all going to diffuse from these endothelial cells back to the smooth muscle cells. So it's a very small molecule that can easily get through cell membranes, okay, and it's going to diffuse from the endothelial cells all over the place in this tissue, and some of it will make its way into the smooth muscle cells. Okay, now what is it going to do then in the smooth muscle cells? Okay, well, it is going to activate an enzyme known as soluble guanylate cyclase. Okay, so let's just talk about soluble guanylate cyclase. So for short, soluble guanylate cyclase is abbreviated as SGC. Okay, and if I draw a smooth muscle cell here, soluble guanylate cyclase is going to be free in the cytoplasm of the cell, hence why it is called soluble guanylate cyclases. Okay, there are other types of guanylate cyclase which are membrane bound. Okay, so notably the receptor for atrial natriuretic peptide. So this is a soluble guanylate cyclase. Okay, right. So what we want to do now is look at the structure of soluble guanylate cyclase so that we can understand how nitric oxide is going to activate it, and then we'll look at the reaction that it's going to catalyze, which you might be able to guess is the generation of cyclic GMP from GTP, and then we'll see what that cyclic GMP is going to do within the smooth muscle cells. Okay, right. So let's firstly start off then with the structure of soluble guanylate cyclase. So soluble guanylate cyclase enzymes are heterodimers. Okay, they're made up of 2 separate subunits, that's what dimer means. Okay, and each of the 2 separate subunits are different, hence heterodimer. Okay, so if I draw these proteins schematically, so I'll draw their primary structure here. So this is one of these proteins from amino terminus to C terminus, and it's going to dimerize with another one that's slightly shorter here. Okay, and these are the 2 separate subunits of a soluble guanylate cyclase. Okay, so the longer one that's on top here is called the soluble guanylate cyclase alpha subunit, and the shorter one that's on bottom here is called the soluble guanylate cyclase beta subunit. Okay, now let me talk now about the different domains of these soluble guanylate cyclase subunits. Okay, so the domains at the C-terminal, which I'm now going to colour in blue, these are what are known as the catalytic domains. Okay, and these are going to dimerize together to make the actual functional enzyme. Okay, so these are the catalytic domains, and these dimerize together to make the actual functional enzyme. So contrast this to the eNOS enzyme. In the eNOS enzyme you had this dimerization which resulted in 2 separate functional enzymes. In the soluble guanylate cyclase you have this dimerization which just results in 1 functional enzyme. Okay, so you need both of these to dimerize together to produce a functional enzyme. Okay, right. Then upstream of the catalytic domain you have a little portion that's alpha helical in nature. Okay, so I'm just drawing it in turquoise and in a spring sort of shape. So this is an alpha helical region here. So this is the alpha helical region. And then further up from the alpha helical region you have a domain known as the PAS domain, which is very important in strongly holding these 2 subunits together. Okay, so these domains are known as the PAS domains in orange here. So each one of the two subunits, the soluble guanylate cyclase alpha subunit and the soluble guanylate cyclase beta subunit, has a PAS domain, and they are very powerfully joined together, and they're very much so involved in holding the two subunits together to form the full soluble guanylate cyclase. Okay, and then upstream of the PAS domain, you then have a domain that's known as HNOX. Okay, so this is the HNOX domain. And in the soluble guanylate cyclase beta, but not in the soluble guanylate cyclase alpha, the HNOX domain has a prosthetic heme group. So here, in the soluble guanylate cyclase beta subunit, this square structure that I'm showing here in turquoise, this is another heme group, okay, which will have an iron cation at the center there. Okay, and the idea here is that nitric oxide, which is diffusing into the smooth muscle cells that's being produced by the endothelial cells, can now bind to that iron cation of this heme group, and that then leads to the activation of the soluble guanylate cyclase catalytic domain down here. Okay, so the binding of the nitric oxide to the heme group leads to a conformational change that then activates the soluble guanylate cyclase catalytic domain. Okay, right. Now, once the catalytic domain is active, what it actually does is it converts GTP, guanosine triphosphate, into cyclic guanosine monophosphate, cyclic GMP, and then also pyrophosphate. Okay, so just like adenylyl cyclase enzymes convert ATP into cyclic AMP, guanylate cyclase enzymes convert GTP into cyclic GMP. Okay, and we will see the adenylyl cyclase pathway later on when we come on to discuss prostacyclin. Okay, right. So this cyclic GMP that's being produced in the vascular smooth muscle cells in response to this nitric oxide, what's it actually going to do? Well, it's going to activate a protein kinase that is known as cyclic GMP dependent protein kinase. Okay, but it has a more more seductive name, shall we say? What have I missed off? I've missed off the protein kinase, cyclic GMP dependent protein kinase. Its other name that students generally prefer is protein kinase G, PKG. So this is protein kinase G. Okay, and it has a very similar reaction profile to protein kinase A, especially in how it's activated. Its activation is very similar to protein kinase A, as you would expect, since protein kinase A is activated by cyclic AMP, and protein kinase G is activated by something very similar, cyclic GMP. Okay, so let me show you the structure of the cyclic GMP-dependent protein kinase. Okay, right. We'll start off with the catalytic domain here, which is the domain that's actually capable of phosphorylating proteins. Okay, and this is at the C-terminal end of the protein. Then, following the catalytic domain, what you have is two regulatory domains here, which are the domains which are capable of binding cyclic GMP. Okay, so here are the two separate regulatory domains. So I might color code this as I go along. So these are regulatory domains. Okay, and the first regulatory domain is known as regulatory domain A, and remember the first one is the one that's closer to the amino terminus, which is this one here. And then the second one is known as the regulatory domain B, which is the one closer to the C terminus, and I'll colour both of them in orange here. Okay, so there are both of the regulatory domains. Then we've also got the catalytic domain down here in turquoise. Okay, right. Then, after the two regulatory domains, you then have a portion that is an autoinhibitory domain here, okay, which is usually sitting in the active site of the catalytic domain and inhibiting it. So this is another example of an autoinhibitory domain inhibiting the catalytic domain, as we saw earlier with myosin light chain kinase. And then finally, you have a special domain further towards the N-terminal than the autoinhibitory domain, which is known as the dimerization domain, or DD for short. So what's thought to happen is two of these protein kinase G enzymes can dimerize together by their dimerization domains binding. Okay, right, so this is the dimerization domain. Okay, so what then happens to activate the protein kinase G is simple. Two cyclic GMP molecules come in and bind one into each of these binding sites on the regulatory domains, okay, and that causes a conformational change in the protein kinase G enzyme that removes the autoinhibitory domain from the active site of the catalytic domain, and the catalytic domain is now active. Okay, right, now, so overall, nitric oxide has activated protein kinase G within these vascular smooth muscle cells. What then is the effect of activating protein kinase G? Well, protein kinase G can go off and phosphorylate myosin light chain kinase. Okay, so this can go and phosphorylate myosin light chain kinase. So if I draw the myosin light chain kinase back again, okay, we'll just draw the catalytic domain here of myosin light chain kinase. Okay, what's going to happen is it's going to stick a phosphate group onto the myosin light chain kinase, and what does this phosphate group do? It stops myosin light chain kinase being activated, okay, even if there is a calcium signal present. Okay, so remember, how was myosin light chain kinase activated? If I get this picture back again, okay. Is it here? No, it's not on that one. On this one, I think, yeah. How was myosin light chain kinase activated? It required the calcium calmodulin complexes to bind to the autoinhibitory domain to move the autoinhibitory domain out of its catalytic site. Basically, what this phosphorylation by protein kinase G does is it means that even if calcium calmodulin complexes are produced, because a calcium signal is produced in the smooth muscle cell, then you're not actually going to get the activation of myosin light chain kinase. So basically, phosphorylation of myosin light chain kinase blocks the activation of myosin light chain kinase by calcium calmodulin complexes. Okay, so it stops smooth muscle cells from being able to contract, quite simply, because remember, myosin light chain kinase activation is what's necessary for smooth muscle cell contraction, since it is myosin light chain kinase that phosphorylates the regulatory myosin light chain and begins cross-bridge cycling. So this is how, well, this is one of the major mechanisms by which endothelial cells all the time are stopping vascular smooth muscle cells in the arteriole, which supplies that local area of tissue, from contracting. Okay, so we'll call it there for this video, and in the next video what we will do is we'll look at another mechanism of endothelium-dependent vasodilatation, which is prostacyclin.